Hello guys, let me introduce myself. My name is Shubham and I'm going to teach you biology today. Today's topic would be biotechnology, and it's basically the unit 9 in CBSE's NCERT book of biology. It consists of basically two chapters, chapter 11 biotechnology principles and processes, and chapter 12 biotechnology and its application. Let's start off. See, you do not need to know about this, but I'm gonna give you like a brief about who Herbert Boyer is. Herbert Boyer is basically a scientist in the University of California, San Francisco, and he was famous for his work with the E. coli and stuff. He is the one who, you can call him the father of biotechnology. He's not actually, but you can call him that because he was the first guy to notice that we can actually code the genetic material in some living organism. Like, he started off with the E. coli bacterium, especially useful properties. So he observed that with enzymes and stuff. You need not to know about that, so I'm not going to elaborate on that as well. So yes, let's start off. Chapter 11, Biotechnology, Principles and Processes. So in this chapter, we're going to learn about principles of biotechnology, tools of recombinant DNA technology, processes of recombinant DNA technology. So what is biotechnology? I'm going to give you the straight definition of European Federation of Biotechnology. I'm not going to give you biotechnology's definition according to the NCERT's book because it's trash. So let's go. It's actually given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB, which is basically European Federation Biotechnology, is as follows: the integration of natural science and organisms, cells, part thereof, the molecular analogues of the products and services. Principles of biotechnology. Among many, two core techniques that enable birth of the modern biotechnology are genetic engineering and bioprocess engineering. I'll elaborate both the things for you. So starting off with genetic engineering. Techniques to alter the chemistry of the genetic material of the diribonucleic acid and the ribonucleic acid is basically known as genetic engineering. So how does it work? You must be thinking, how does the genetic engineering works? Some people are eager to study genetic engineering as well as a course in the bachelor's. So yes, I'm gonna tell you how and who told you to how to do this, and do not try this at home because you do not have the proper instruments and stuff. So yeah, genetic engineering is basically coding away according to your own with the DNA and RNA. We basically modify it. Okay, so every cell has DNA and RNA. Basically the genes, in simpler term, in layman terms, you say genes is basically made up of this stuffs. So DNA or diribonucleic acid can be modified. See, the genetic makeup, if you can modify someone's genetic makeup, think about it. We can design anything. We can design super soldiers. We can design workers who would not even stop working. We may not there would not be any defective piece. You must have seen in universe, there are people who are crippled, who are who are not that good, like people with Down syndrome, people with Turner syndrome, people with Kwashiorkor, people with obesity, people with elephantiasis, with many diseases and stuff. So they basically are not perfect like any other normal human being, like you and me. Okay, so that does not make any sense. Do they deserve that? Absolutely not. Who deserves that? So for that reason only, for that particular reason, we have been developing genetic engineering. See, what if the newborn baby, before even the birth of it, we would like to genetically modify it. If a mother wants her kid to be tall, the kid would be tall. Does not matter if the mother and father is dwarf. See, the offspring, the genetic makeup of offspring basically is like the inheritance and some mutation. Mutation is a different whole other thing. I'll teach you that in a separate video. So let's not talk about mutation right now because it's so low in mammals and reptilians also. It's basically in viruses and stuff. Virus mutates more than they reproduces. So yes. So, well, basically, genetic engineering, you got the idea, right? What genetic engineering actually is. So, it's nothing basically just to modify DNA and RNA to get the desirable set of traits. So, yeah. So, I'm going to talk about bioprocess engineering, maintenance of the sterile, microbial contamination free. Sterile means like disease free, basically, in easy words. Ambience in chemical engineering processes to enable growth of the only desired microbe, eukaryotic cell in particular, large quantities for the manufacture of the biological products like antibiotics, vaccines, and enzymes, etc. See how a vaccine is made. Oh, well, it's basically either the dead virus or a living virus. Okay, which is basically weakened. Like talk about COVID vaccine, like must be COVID Shield, Pfizer, Moderna, Johnson & Johnson, does not matter what it actually contains. Either it contains the dead virus or the semi-living virus, which will not harm you, but the human body will create its own antidote against it, because that's how biology works, that's how metabolism works, that's how vaccination works. Body will think that we are suffering with the disease and will continue to produce antibodies to fight against them. Okay, so that's how it works. So it's basically genetically engineered bacteria or viruses are modified to create vaccines, enzymes, and antibiotics to fight off disease. So like, how you can tell about the gene transfer and gene cloning, recombinant DNA? You must think that these are big words, but that's not the point. I'm going to teach you bit by bit. Okay, you probably appreciate the advantages of the sexual reproduction over asexual reproduction. You know that we just saw that in the last chapter. Okay, so basically the multiplication of undesirable genes along with the desirable ones, the techniques of genetic engineering, which include the creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation. Okay, so like using these techniques we can overcome any disparity in human being, or not human being, any living organism. So like origin of replication is terminal. See, cloning, what cloning is, I'm going to give you a brief idea about cloning. Okay, so well, cloning is nothing but to make a copy itself, like exact copy. Cloning is not reproduction. Hydra clones. Hydra does not reproduces, it clones. See, cloning is just like making a copy, like making a rip-off of you, and it's not your offspring, it's basically the same genetic material. It's like more of a twin, not a copy, I guess. Yeah, so making multiple identical copy of any template of DNA, that's called cloning. So let's now focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid. What a plasmid? It's basically autonomously replicating circular extra chromosomal DNA. Salmonella typhimurium, it's basically the bacteria, 1972. You need not to remember that because that's not going to come. Molecular scissors. See, basically molecular scissors is restriction enzymes, and restriction enzymes is otherwise known as molecular scissors. They cut the DNA in pieces, and that were basically linked with the plasmid DNA. So it might come 1 mark question in CHSE exams. What are molecular scissors? You can tell that it's restriction enzymes, and they can ask you that in Viva too. So yeah, okay. The cut piece of a DNA was linked with plasmid DNA. These plasmid DNA act as vectors. Vectors basically are the pieces of the DNA that are attached to it and can be transferable. You probably know that mosquito acts as insect vector. Like vector is someone who carries things, okay? Like mosquito carries malaria to us. It basically like whole stage, okay? So yeah, E. coli was called cloning of antibiotic resistance gene in E. coli. Like if we successfully clone the antibiotic gene in E. coli, we could use it for antibiotics for fevers and many other common cold disease like common cold and stuff. So yeah, sorry about that. Okay, so identification of DNA with basically you can hence infer that there are three basic steps in genetically modifying an organism: identification of the DNA with desirable genes, introduction of the identified DNA into the host, maintenance of introduced DNA in the host, transfer of the DNA into its progeny. Okay, I'm gonna teach you that bit by bit. Identification of DNA with desirable traits. There's millions of billions of trillions of phases in a DNA. Like how you gonna, like it's so small and it's literally in the every cell of a human being. Like okay, you talk about chromosome, like, but how are you gonna know that this is it, this is a trait we want, this is the gene that controls the eye color, this is the gene that controls the skin color? It can be multiple genes, it can be one gene, like basically reason for many traits. Okay, one gene may be controlling eye color and hair color, and one gene may be just responsible for the face color. Okay, how much melanin present in you. Okay, so that's it. The first step is that to modify an organism is just to identify what particular genes is that in that particular DNA, what trait we need. Okay, so introduction of the identified DNA into the host. See, you cut off the DNA from another bacteria, like bacteria, E. coli bacterium for in this case as example. Okay, so it's basically you dig up whole thing you need from the host cell. Again, like the host cell is someone who hosts things. The literal meaning of it is like host things. Like you go to your some distant relative, they are the host, like the one house you are living in. Like same in this in biological concept also, DNA gonna go to a host. Like the set you cut off the DNA who is basically reason for hair color, like or eye color for example, you put that in another host. Host means in that particular body you want the change in. Okay, in host is the one you want the change to be followed. Okay, so in that host would be a new organism, like whole other traits, like manipulated organism. So maintenance of the introduced DNA in the host and transfer of the DNA to its progeny. Okay, so the thing is that maintaining the introduced DNA, what does that means? Okay, so these things are very delicate and small. Okay, as these things are very delicate and small, so we cannot take any chances. Okay, if we take any chances, the thing is that we might screw up, and if we screw up, then it's gonna be a problem. Okay, so just leave it at that. These things are to be done with high precision. Okay, so tools of the recombinant DNA technology. Restriction enzyme. Well, the restriction enzymes are responsible for restricting the growth of the bacteriophage in Escherichia coli, which is basically E. coli, Escherichia coli. Okay, so one of these added to methyl groups of DNA. See, E. coli was isolated from the bacteriophage and basically were added into the methyl groups of the DNA, while the other cut DNA, that latter was called restriction endonuclease. Remember the term restriction endonuclease, guys. The first restriction endonuclease HIN2, whose functioning depended on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. It was found that HIN2 always cut DNA molecules as a part of point by recognizing the specific sequence of 6 base pair. This specific base sequences is known as the recognition sequence for HEN2. Besides HEN2, today we know more than 900 restriction enzymes. Remember that's gonna come in NEET. That 900 restriction enzymes have been isolated over literally 230 strains of bacteria, each of which recognize different recognition sequences. 230 strains of bacteria, each of following recognize. Okay, so see, E. coli, how the naming comes. If so many enzymes, how are we going to name it? So the conventional naming for this enzymes is first letter of the name comes from the genus, and the second two letters comes from the pardon me for that, sorry, that was my friend. So two letters come from species of the prokaryotic cell which they were isolated. Example, ECORI come from the Escherichia coli RY13. In E. coli, the letter R is derived from the name of strain. Roman numbers following the names indicate the order in which the enzymes were isolated from the strain of bacteria. Okay, so restriction enzymes belong to a larger class of enzymes that are called nucleases. These are two kinds: exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of DNA, whereas endonucleases make cuts at specific positions with DNA. Each restriction endonuclease functions with inspecting the length of the DNA sequence. Once it finds specific recognition sequence, it will bind to the DNA and cut with each two strands of the double helix at specific points in their sugar phosphate backbones. In figure 11.1, you can see that also. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in DNA. Okay. See, we're gonna learn about this whole cutting of the DNA. The enzyme cuts DNA strand at same time. Look at this. This is vector DNA. The red one is the vector DNA. E. coli cuts the DNA between bases G and A only when the sequence G, A, T, T, and C is present in the DNA. See, the G and A is only cut. Look at that. Really look at that. See, G in the second figure, you can see that GC is there, TT is there, AA is there, and the end of it is the sticky end. A sticky end is basically that particular end which is gonna get joined. Okay, the sticky end of the opposite host DNA and the foreign DNA, that sticky end is connected. Both sticky ends are connected and they're joined. DNA fragments join at the sticky end. That's what I've been telling you about. So, and in the last figure, you can see the recombinant DNA or the mutated or the modified gene is here, like the recombinant DNA in here. Recombinant DNA is basically the new, like the modified DNA we get, like isolating the whole separate genes, like the suitable traits we need from the organism, and putting that into the host body. The new organism would be called as the new organism. Now it would be wrong to say that. The new DNA, okay, the new strand of DNA would be known as recombinant DNA. So basically this is the steps in formation of the recombinant DNA by action of restriction endonuclease enzyme E-coli. So you guys must remember what E-coli is, alright? Escherichia coli RY13, okay? So yeah, like actually I'm gonna teach you how to know that what exact direction are we gonna cut, okay? So reading the kept in mind, for example, following sequences reads the same on two strands, like 5' to 3' direction, okay? So in the following sequences read the same on two strands, 5' to 3' direction. This is also true if 3' to 5' direction, which is basically if A is equals to B, then B is also equals to A. Okay? See, if 5' is G, A, T, T, and C, then the reverse would also be C, T, T, A, and G. Okay, see GAATTC, and the reverse, like basically 5 dash to 3 dash. If we're gonna go from 3 dash to 5 dash, is basically CTTAAG, which is the reverse of the opposite of it. Okay, so yeah, restrictions enzyme cut the strand of DNA a little away from the center of the palindromic sites to give them positions. Okay, two bases of opposite strands, basically they leave around a sticky end so that they're gonna stick up. Okay, they're gonna get joined and form a recombinant DNA. See, you see this DNA ligase, basically foreign DNA. This blue colored DNA strand, diribonucleic acid strand, is basically a foreign DNA, and this red one, which is always closed, and the plasmid DNA is always closed. Remember that it's always closed. It's not open-ended. Not unlike the foreign DNA, it's not open-ended. Vector DNA plasmid, same restriction enzyme cutting both foreign DNA and vector DNA at the same point. So ligases join foreign DNA as a plasmid. Look at that. Will you look at that? I'm going to zoom it up. Recombinant DNA molecule, and the transformation is this. See the transformation? You guys are seeing it properly, like a diagrammatic representation of the recombinant DNA technology. So that's how it happens. Vector DNA, we remove a part of it, the part we do not need it, and we have isolated another specific trait from our foreign DNA, and we cut it down, put it down in the vector DNA plasmid, so that ligase join foreign DNA to plasmid, and then recombinant DNA is formed. This is known as the recombinant DNA. Okay, all right. So the transformation is like E. coli cloning. It, as you know, that bacteria clones. So cell divides and it clones. Okay. So yeah, like the separation and isolation of the DNA fragments. Okay, the cutting of DNA by restriction endonucleases results in fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. Gel electrophoresis is important. I'm going to tell you later why it's important. Since DNA fragments are negatively charged molecules, they can be separated by forcing them towards the anode under electric field. Those a medium of matrix. See, anode attracts the negative. Okay, we know that in physics and chemistry as well. So nowadays most commonly used matrix is Agarose. Matrix is basically a medium in which anode is placed so that the negative charged particles, which are DNA, is going to go to anode. Okay, so under electric field, though, that medium or matrix is basically agarose. Remember that, agarose, which is natural polymer extracted from seaweeds. Seaweeds are basically small sea creatures. You can Google it up. Okay, I'm not going to waste my time teaching you that. The DNA fragments separate, resolve according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. Look at figure 11.3. Okay, see, type of agarose gel electrophoresis showing migration of undigested and digested set of DNA fragments. It's basically from line 1, then line 2 to 4 respectively. See, if this whole white box, what is this? This is a medium or a matrix. See, number 1, 2, 3, 4 are the wells, and DNA bands are the red ones, basically open-ended DNA chains. The separated DNA fragments can be visualized only after staining the DNA with both compound known as ethidium bromide followed by exposure of UV radiation. You cannot see pure DNA fragments visible of light without staining. So yeah, so cloning vectors. So how about cloning vectors? After the vector is formed, like how can we clone it? It's not just one copy, we need like tons of it, millions and billions of it. Okay, how to make it? So you know the plasmids and bacteriophages have ability to replicate within bacterial cells independent of control of chromosomal DNA. Bacteriophages, because of their high number per cell, have very high copy numbers of the genome within the bacterial cells. Some plasmids may have only 1 or 2 copies per cell, whereas others may have 15 or 100 copies per cell. Their numbers can go even higher. if we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its numbers equal to the copy number of the plasmid or bacteriophage. Vectors used at present are engineered in such a way that they help easy linkage of the foreign DNA and selection of recombinants from the non-recombinants. Okay, so the following features that are required to facilitate cloning into a vector is origin of replication arrays, selectable marker, cloning sites, and that's it. So vectors for cloning genes in plants and animals, we're gonna learn about that earlier. First we're gonna talk about origin of replication. This is a sequence basically from by which where the can be like replication starts. It's the first place in which the replication starts. Okay, so the piece of DNA when linked to a sequence can be made to replicate within the host cell. The sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to recover many copies of the target DNA, should be cloned in a vector whose origin of support high copy number. Okay, so origin of replication is basically the first place it gets copied in. Okay, the sequence is get copied. So if you gonna see a vector with a small open chain and not a lot of space to copy, so it's up to you. You choose if you like, you need a small vector or a bigger one to need more copies for it. It's up to you. So selectable marker, sorry for that. In addition to ORI, the vector requires a selectable marker, which helps in identifying, eliminating non-transformants, selectively permitting the growth of transformants. So see, non-transformants is basically, as say in gene, these are very delicate processes. Like these are the very delicate things. Sorry for that again. Sorry guys, sorry fellas. So yeah, so as we know that this has been like a very delicate process, we're gonna be like that. So if it's delicate, so there are chances of errors. If there are chances of errors, so all we need to do is to get an extra. So if we need a 20 of it, so we take 100, so that 80 would be waste and 20 we can take. Okay, so transformation is a procedure through which a piece of DNA is introduced in a host bacterium. You will study the process in subsequent section. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramiphenol, and tetracycline or kanamycin, etc. So I'm going to repeat that again. Okay. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramiphenol, tetracycline, and kanamycin. Remember the name: ampicillin, chloramiphenol, and tetracycline, and kanamycin, etc. Okay, are considered as useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against these antibiotics. Okay, so cloning site. In this figure, we're gonna see that cloning site. Okay, I'm gonna talk about it later. In order to link the alien DNA, the vector needs to have very few and preferably single recognition sites for common use restriction enzymes. Presence of more than one recognition sites within the vector will generate several fragments, which will complicate the gene cloning. The litigation of alien DNA is carried out as a restriction site present in one of two antibiotic resistance genes. For example, you can litigate a foreign DNA at BAM1 site of tetracycline. The resistance gene would be vector of PBR322. Just remember that, PBR322. Alright, the recombinant plasmids will lose tetracycline and resistance due to insertion of foreign DNA, but can still be selected out of the non-recombinant ones by plating the transformants on tetracycline containing medium. The transformants growing on ampicillin containing medium are then transferred on a medium containing tetracycline. Okay, the recombinants will grow in ampicillin containing medium but not on that containing tetracycline. but the non-recombinants will grow on the medium containing both the antibiotics. In that case, one antibiotic resistance gene helps in selecting the transformants, whereas the other antibiotic resistance. Okay. So in the next class, we're gonna learn about vectors for cloning in genes and plants and animals. That's it for today, guys. Do not forget to like, subscribe, and share this channel. And if you like want more of me to teach you, like if you got any doubt, you can personally contact me on Instagram. It's basically @ig.weed. It's ig.we3d. You can follow me on Instagram, and do not forget to like and comment also your doubts in the videos, and I'm gonna try to clear it up. Okay, I'm gonna try to reply. Okay, and do not forget to subscribe, like, and comment. Thank you guys. See you in the next video.